of her. Julia did her PhD in Myrtle Rust in New Caledonia, combining ecology, genetics, and plant pathology. That sounds like a monster PhD to me. She is now a forest pathologist at Scion, based there in the Rotorua office. I'm sure her PhD has her very well qualified for that, and she did a postdoc there as well. Right, Julia, I think we should be ready to go. I can see your screen there. You can take it away.
Yeah, awesome. Thank you, Renee. So as you said, Renee, I'm a forest pathologist at Scion. Oh, sorry, I can just need to hide this up. And I've been working on Myrtle Rust since a few years now, doing my PhD in New Caledonia and doing my postdoc in New Zealand. And I've been mainly working on evaluating how plant diseases are impacting different host species, species, and on ways to mitigate this impact by using, for example, selection for resistance. So today I will be talking about the different strains of Myrtle Rust and why do they represent a biosecurity threat for New Zealand and for all countries that have a rich biodiversity of Myrtaceae plants. So COVID-19 has taught us a lot about pandemics and epidemiological terms, and we can actually draw some parallels between pandemics in plants and the COVID-19 situation. For example, both humans and plants are attacked by pathogens. They can be virus, bacteria, or fungi. And in both cases, the disease will occur when the pathogen will meet the susceptible host and the favorable environment. This is a concept called the disease triangle, and it is mainly used in plant pathology to predict epidemic outcomes. In nature, for humans and plants, there is a constant evolutionary battle between the pathogens and their host, and each continuously evolve to overcome the other. So, for example, when the host will express some degree of resistance, the disease triangle will result in a lower level of disease. And to overcome that, the pathogen will evolve and new strains will arise that are able to infect the resistant host. So by definition, a strain is a genetic variation that is created by sexual recombination or mutation. And a mutation, it's a change in the DNA sequence that in that case could be beneficial to the pathogen. The strain of a pathogen will be different from the original strain, and so it will behave differently. For example, everyone heard about the detection of several strains of COVID-19 emerging in the UK, in the US, or in Brazil. And those strains have a large number of mutations that explain why they spread so easily and quickly. The same principle applies to Austropuccinia psidii, which is the causal agent of Myrtle Rust. And for the rest of this presentation, I will refer to it as Myrtle Rust. So today I thought I would talk to you about how many strains of Myrtle Rust exist in the world and where do they occur. We are going to discuss the potential impact of introduction of new strains of Myrtle Rust in New Zealand. And finally, we will verify if all the strains of Myrtle Rust can infect New Zealand species. So this is a brief intro for Myrtle Rust. So it is a fungal pathogen native from South America and causing the rust disease on Myrtaceae plants. It has a very broad and unusual host range for rust fungi. So far, at least 480 species are infected by this pathogen worldwide. The disease affects the young and expanding foliage tissue, the flowers, and the fruits. And when the infection is severe, it can lead to the plant death. You can easily recognize the disease by the yellow pustules that are resulting from the infection. These pustules contain yellow asexual spores that are called uridinospores. These spores are airborne and easily dispersed by the wind, by the humans on your clothes or on your cars, and also by the wildlife. So on the bottom left, it's a funny picture that I've taken in New Caledonia, and it's showing you a lizard covered with Myrtle Rust spores, and it was going from one tree to another and just spreading Myrtle Rust all over the place. So there are several strains of Myrtle Rust existing in the world, and each of these strains have specific distribution and host range. In 2017, Soewarto et al. Analyzed the genetic diversity of Myrtle Rust in the Americas and Hawaii. They have identified 9 genetic clusters among 200 isolates of the pathogen, and they have assessed the genetic relationship among these clusters, which is showed here on the minimum spanning network. And they have demonstrated distinct subnetwork or biotype associated with these genetic clusters. So there are at least 4 of these biotypes that have been identified. One of these biotypes comprised the cluster C1, C4, which include isolates that are able to infect a wide variety of host species, and which is also a biotype that have a wide distribution worldwide. This biotype is referred as the pandemic biotype, and it is the one that is found in Australia, in New Zealand, and in New Caledonia. The second biotype is comprised of the cluster C2 and C3, which infect eucalyptus species and Syzygium jumbos in Brazil and Uruguay. And finally, there is a biotype that is very distinct and associated with the guavas in the guava species, in Brazil, and it is the cluster C6. There is another biotype. Here we go. So it's the biotype C8 associated with the species Pimenta joica in Jamaica and the native Myrinium in Uruguay. And additionally, there is another strain of Myrtle Rust found in South Africa that is unique, different from the pandemic strain and from all the other strains identified so far from South America. So this South African strain is able to infect several different species of Myrtaceae. So Myrtle Rust occurs widely across the globe, and it is now recognized as one of the most invasive forest disease. It is the cause of serious problems in Myrtaceae-based industries, and it is also threatening the biodiversity. This map shows you the initial geographic distribution of Myrtle Rust since its discovery on guavas in Brazil in 1884. It is assumed that the rust is originated from this region, and it was probably present in South and Central America prior to be reported in publications. As explained earlier, there are multiple strains of the pathogen occurring there. From 1997, one pandemic strain has really begun to take off and start invading new countries, starting with Florida, California, Hawaii, the Southeast Asia, Australia, New Caledonia, and finally New Zealand in 2017. The host range of this pandemic strain already comprises several hundreds of species and is constantly expanding. In 2013, a strain only found in South Africa have been detected and able to infect both native and exotic species. So we know what the current situation with the pandemic strain of Myrtle Rust in New Zealand is, But our concern is that we don't want the older strains to arrive. And just for a reminder, the host range of the pandemic strain in New Zealand comprise 30 host species. The pandemic strain is distributed throughout the North Island and parts of the South Island, so quite a wide distribution throughout the country. Despite many species being susceptible, artificial inoculation studies with the pandemic strain has demonstrated that some species were expressing some level of resistance to the pandemic strain, such as the mānuka species and a couple of Kunzea species. On the other hand, the same artificial inoculation study showed no resistance for species like Metrosideros excelsa and low-formator species. In the field, we are only starting to understand the impact of the pandemic strain on highly susceptible species. Low-formator species in particular, they are heavily impacted on their fruits, on their flowers, and at the seedling stage, which will result ultimately to these species to disappear from their natural environment. And we know that such a scenario is likely to happen because it is also what Australian scientists have reported with some of their species. So the risk associated with the introduction of new strain of Myrtle Rust in New Zealand, they could increase the host range, they could overcome the host resistance, change the aggressiveness of infection, reduce the effectiveness of management tools. So serious impact of new strains of Myrtle Rust being introduced in areas already infected by the rust occurred in the past. And one of the most famous examples comes from Jamaica with the allspice industry, which is also known as the Jamaican pepper. So back in the early 90s, a strain of Myrtle Rust was present in Jamaica and only infecting Syzygium species. This was not really a problem since Syzygium species were exotics and it was not causing any economical impact. The problems really started when a second strain of Myrtle Rust was introduced in 1934, and this strain was seriously infecting the pimenta species, in particular the species cultivated for the allspice industry. And within just a couple of years following the introduction of this new strain, the whole production of allspice collapsed. So to avoid this type of scenario to happen in New Zealand, we need to prepare. We need to better understand the potential threat that this other strain of Myrtle Rust occurring in the world represent for New Zealand. And to do this, New Zealand has teamed up with a network of scientists from where these new strains could come from. And here is the result of one of these collaborations. So with scientists from FABI in South Africa, we have tested the susceptibility of native New Zealand Myrtaceae to the South African strain of Myrtle Rust. So for this testing, seeds of 4 native Myrtaceae were collected from different sites across the North Island and sent to South Africa following strict hygiene and permit conditions. So the 4 species were Kunzea robusta, also known as kanuka, Kunzea linearis, mānuka rawiri, Leptospermum scoparium, mānuka, and Metrosideros excelsa, pōhutukawa. So in South Africa, the seeds were germinated and ground to a size appropriate for being artificially inoculated. The inoculum, which is a suspension of the yellow spores, originally came from a single pustule isolate that has been built up on a very susceptible host. So the seedlings of each species were inoculated with the recommended spore concentration and following a protocol to maximize the infection. the fungal infection has been monitored and final disease assessment was made at 14 days after inoculation. We have repeated the inoculation after pruning the seedlings and after leaving them enough time to grow new healthy tissue. So in the subsequent analysis, I will call them inoculation 1 and inoculation 2. The final assessment comprised the following variables, so disease incidence, the stem infection incidence, and the leaf severity, which is a visual estimation of the percentage of the leaf area covered by the spores. And here I would just like to acknowledge the efficient and welcoming team that helped me to carry out the disease assessment in South Africa, especially Irene, Esna, Gina, and Louise. So what we have observed is that the 4 New Zealand species that we have inoculated were susceptible to the South African strain of Myrtle Rust. Pustules containing spores were developing on young leaves and on the stems as early as 7 days after inoculation. By 14 days, dieback of the growing tips started to appear on all the species because of the rust infection. On Leptospermum scoparium, flowers and flower buds were infected every time that they were present, and this resulted in flower abortion. So while all the species were susceptible, they actually showed some differences in their susceptibility. For example, if you look at the bar plot here with the disease incidence, which is the proportion of susceptible seedlings, you can see that the variation is between the species and also between the inoculation, so inoculation 1 and inoculation 2. And this difference were confirmed by a multi-comparison test for at least 3 of the species. We could not include Kunzea linearis in this test because of the insufficient data available. So in the statistical test, a significant difference will be shown by lower letter cases. So overall, what this test shows is that after inoculation 1, Metrosideros, excelsa, and Leptospermum scoparium were not significantly different from each other, with around 70% of susceptible seedlings. But the disease incidence for these 2 species was significantly higher than Kunzea robusta, which only had 80% of its seedlings being susceptible. After inoculation 2, Leptospermum scoparium became the most infected species with around 98% of all the inoculated seedlings that were infected, followed by Metrosideros excelsa, 74%, and Kunzea robusta, 71%. So interestingly, we observed that there was no significant difference in the disease incidence for Metrosideros excelsa between the 2 inoculations. So that means we approximately had the same seedlings being infected every time. But for Leptospermum scoparium and Kunzea robusta, we had a higher disease incidence after inoculation 2. So although Kunzea linearis was excluded from the statistical test, we can still see that the disease incidence was quite high, between 60 and 65% of the seedlings were susceptible across both inoculation. And another point that I'd like to highlight is that we also had seedlings that were constantly free of symptoms after each inoculation. It is unlikely that these seedlings would have escaped the disease twice, so they could be potentially resistant. So we have identified 30% of these seedlings in Kunzea robusta, 3% in Metrosideros excelsa, and 1% in Leptospermum scoparium. So we did the same analysis with the disease severity on the leaves, and again we had significant differences between species and inoculation. So after inoculation 1, Metrosideros excelsa was the most severely impacted species with 54 of the leaf area covered with spores, followed by Leptospermum scoparium, 31%, and Kunzea robusta, 5%. After inoculation 2, Leptospermum scoparium became the most severely impacted species with, on average, 80% of the leaves area covered with spores. No significant differences were seen between Metrosideros excelsa and Kunzea robusta, which were around 35% of leaf area covered with spores. Again, the disease severity did not significantly change between the two inoculations for Metrosideros excelsa, but for both Leptospermum scoparium and Kunzea robusta, we had a higher disease severity after inoculation 2. And for Kunzea linearis, the disease severity was 22% after inoculation 1 and 34% after inoculation 2. For the stem infection, we were only able to collect the data after inoculation 1, but we saw already differences between the species. So for both Metrosideros excelsa and Leptospermum scoparium, we had significantly more stem infection than Kunzea robusta. We have recorded between 30 and 45% of the seedlings showing infection on the stem for Leptospermum scoparium and Metrosideros, and only 8% for Kunzea robusta. And just for information, Kunzea linearis had 41% of seedlings with stem infection. Another findings from this work comprise the timing of when particular type of spores were seen on each of the species. So spores, so the yellow type of spores, were observed at 7 days after inoculation on Leptospermum scoparium, on Metrosideros excelsa, and on Kunzea linearis. The spores were observed a little bit later, at 9 days after inoculation, on Kunzea robusta. By 12 days after inoculation, teliospores were developing on all the species, with Kunzea robusta having the least amount. The teliospores are the brownish type of spores that you can see here on the picture, and they are the precursor of sexual recombination for Myrtle Rust. By 21 days after inoculation, basidiospores, which are the reproductive type of spores, were observed on some of the seedlings, providing further evidence that Myrtle Rust is able to complete its life cycle on a single host. So to summarize the findings, the 4 species from New Zealand that we have tested, Leptospermum scoparium, Metrosideros excelsa, Kunzea linearis, and Kunzea robusta, are all susceptible to the South African strain of Myrtle Rust. Kunzea robusta was the least susceptible species, while Leptospermum scoparium and Metrosideros excelsa were the most susceptible. Similar susceptibility ranking was reported under artificial inoculation with the pandemic strain, and this might indicate similar pathways of infection or host responses to both of these Myrtle Rust strains. We observed severe infection resulting in dieback on the growing tips for most of the species, and on reproductive parts, flower buds, and flowers for Leptospermum scoparium. The host range of the South African strain of Myrtle Rust now includes 21 species, and this includes 4 native species from New Zealand, but potentially more. The South African strain of Myrtle Rust is therefore a biosecurity threat for New Zealand native Myrtaceae and for other Myrtaceae worldwide. The thalia, which are the precursors of sexual recombination, were found readily on the leaves and stem of New Zealand Myrtaceae, following the inoculation with the South African strain. The South African strain of Myrtle Rust can complete its life cycle within 3 weeks on New Zealand Myrtaceae host, demonstrating its adaptative potential. More severe symptoms were recorded on 2 species, Kunzea robusta and Leptospermum scoparium, after we pruned them, and this highlights the importance of plant growth rate and timing of inoculation in determining the vulnerability of the species. And it was important to repeat the inoculations to fully capture the susceptibility. In terms of future work, it will be interesting to repeat this inoculation trial on further New Zealand species and more seeds families. The effect of the environment condition have not been investigated in this study, but it will be interesting to see if the disease expressions change under different environmental conditions. And finally, the research is ongoing to screen the seedlings germinated from the same seed lots with other strains such as the eucalyptus strain in Uruguay. All our work regarding the testing with the South African strain of Myrtle Rust has been published last year, and here the link if you want to access the publication. So the take-home message here is that the introduction of the South African strain of Myrtle Rust to New Zealand could have devastating consequences, similar or worse to those seen following the introduction of the pandemic strain. Continued vigilance and a coordinated biosecurity approach to exclude further strains of Myrtle Rust from New Zealand and from the surrounding Pacific countries is crucial to minimize the ecological and economic impact. I also want to add that COVID-19 taught us a lot about dealing with different strains of the same pathogen, but we must not forget that many plant species are facing the same challenge. Thank you for your attention. So I would like to acknowledge and thanks the Mana Whenua who kindly allowed scientists to do this research. I would also like to acknowledge our different funders and our local and international partners involved in this project.
Thank you very much, Julia. Thank you for that excellent presentation. And we have a number of questions here, so I think we will just carry on with the webinar and see if we can get them answered. I will ask them roughly in the order in which they were asked. Hopefully I can keep on top of that. Were these inoculations repeated on the same plants, or were inoculations 1 and 2 on separate sets of plants?
The inoculation was repeated on the same set of plants. Yeah.
Okay, relatively straightforward answer there. Any feeling on how accurately artificial inoculation results reflect disease levels in the field associated with natural infections?
Yeah, that's a very interesting question. So I think under artificial inoculation, the conditions are optimum for the disease development. So it's pretty much like we observe the worst case scenario of what could happen. But we really need to couple that with experiment in the field. We need to have more indication on all the disease and all the plants will behave under field condition.
It certainly would be ideal. Did you test pathogenicity of the basidios spores on all the hosts?
No, we did not. I think it was a little bit, how do you say, out of the scope of this study, but it would have been interesting to do it. I know that it has been done previously with scientists working at FABI, so I think the paper should be available.
Okay, very good. What was the host plant in your New Caledonia image the one with the lizard? Excellent lizard, by the way. It was very nice.
Oh, yeah, it's one of my favorite. It's called a Genia gacogni.
The lizard or the plant? Okay. What is the level of, well, okay, I'm not sure whether this is really within your level of expertise, but we could have a quick acknowledgement of this one. The level of awareness of the South African strain in Australia, and what action are they taking for their own biosecurity and for our downwind biosecurity? I'm not sure about your awareness of what's going on in Australia, Julia.
Yeah, I'm not too sure, but I believe they would be taking the same approach as New Zealand. So they would be having different layers of control to avoid the strain to enter in Australia. So pre-border control, controlling passengers from planes and things like that.
And we do
Surveillance.
we do have, of course, a two-day, at least part online event coming up in Australia about the situation there.
Yeah.
So people could certainly tune into that if they were interested. Perhaps drop me an email after this if you'd like more information. I can remind myself of exactly when that is coming up. I think it's later this month, right, Julia?
Yeah. I think it's on the 23rd, 25th of March.
Excellent. Right.
And
Do we know if the
I,
host? Sorry, go ahead.
oh, sorry, and just letting you know that research is ongoing to develop a tool that will enable to differentiate the different strain of Myrtle Rust.
It is correct. Now, do we know if the host range differs between the pandemic versus the South African strains? Oh, I think that's probably a tricky one to answer right now. Do you have any comment on that?
No, we don't know because we need more testing.
We certainly
At the
do.
at the moment, we know that at least 4 species, the 4 that we have tested here, are susceptible to the South African strain, and they are also susceptible to the pandemic strain under artificial inoculation. So, yeah, there are some similarities between those 2 strains.
Okay, just a couple more questions here. Acknowledgement, thanking you for such a complete presentation, Julia. Quite terrifying. The question is the following. As Myrtle Rust arrived in New Zealand by itself, it seems that our activities have a little to nothing to do with the introduction of the pathogen or successive strains. And I was wondering this myself, how much do we know about vectors of transmission apart from strong winds, i.e. have there been instances where people moving plants have likely moved this disease between countries?
Yeah, there are many pathways for pathogen in general to be introduced in a country. I guess with the COVID-19 situation at the moment, we don't have much travelers coming from foreign countries, but the wind for Myrtle Rust is probably the worst vectors, you know. And it would be difficult to control the wind, but what we can do is to invest into early detection of new strains, you know, so we can control them quicker and we can avoid that they propagate in New Zealand.
We certainly can. All right, I'll just ask the last question now. What are the implications of being able to complete the life cycle on a single host? Is this the same for all species or individuals? And what influences the completion of the life cycle? So quite a bit going on there. Would you like to start with the first one, the implications of being able to, you just need one host with this?
Yeah, I guess that this ability to complete the entire life cycle on one host to come from the historical belief that rust usually needs 2 hosts to complete a life cycle. So every time that we see basidiospores coming from the same host, it's justification that this rust behave differently and can complete its whole life cycle within just one host. The other implication is that this means sexual reproduction, and this means probably an increase of genotypic diversity. So probably the creation of more virulent strains, for example.
Okay, is it the same for all species or individuals? Well, I guess as far as we know, right?
Oh, right. So we start to have good knowledge about sexual reproduction happening for the South African strain. We know it's happening with the pandemic strain. We know it's happening also in some of the strains in South America, even if the clonal reproduction is the major way of the pathogen to propagate. But I guess we need more studies in that field, and we need really to compare all the different strains together. And I don't think this has been done. I don't think anyone want to risk to have the South African strain and the pandemic strain and one of the South American strain in the same room. That could be maybe too
Yes.
dangerous. I don't know.
Yes.
But
Some
this
security
this
challenges
would be
there.
interesting to investigate.
And as for what influences the completion of the life cycle, I guess it's just again almost disease triangle stuff. It's going to depend on the host, the climate, perhaps even the
Exactly.
strain.
Yeah, more genetic diversity, new strain could lead to this strain being able to infect new host species or infect a new area, having a better temperature tolerance or things like that.
And one last comment just to wrap up. Historically, there are many examples of wind dispersal or rust across the Tasman, but not sure if there's any over longer distances, e.g. from South African. Perhaps humans are needed to be the vector. Just to share that one with the audience. Yes,
Yeah,
yes, indeed. Right, so
yeah.
with that, I'm going to wrap it up. Thank you very much, Julia, and thank you very much
Thank
to the
you.
audience for providing some excellent and very thoughtful questions in this morning's webinar. So as always, a video of the webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and it will also be emailed to everyone who registered. The next webinar will be Wednesday, April 7th, when we will hear from Emma Bodley, Conservation Specialist at the Auckland Botanic Gardens. And her talk is titled Strings Attached, Managing Ex Situ Plants Highly Susceptible to Pathogens. So we're going to hear about all kinds of challenges from her then, I'm sure, and I hope to see you all then.